about that.
No problem.
We didn't miss too much. About 1 year ago, SpaceX's Crew Dragon Demo 2 flew to the International Space Station, and it was about on May 31st last year. And this was a part of the Commercial Crew Program. It was the first commercial system to send humans into orbit, and it was also the first crewed orbital space flight coming from the US since 2011 during the last space shuttle mission. And since then, last November, there was the SpaceX Crew 1, which also went to the International Space Station. And very soon, in just a few days, the Crew 2 will also be heading to the International Space Station. And there's also just plans, if you look on NASA's kind of launch plan of SpaceX's launch plan site, you'd see that there are many more plans for more manned spaceflight missions. And so I don't know if you guys remember this around last year, but when the 2 astronauts were docking onto the International Space Station, one of the astronauts actually bumped his head on, I guess it was kind of like the entrance. And I remember at the time there was just a whole bunch of kind of like clickbaity articles speculating if he had some head injury. But fortunately, everything was totally okay, and it was just like an overinflated issue. But I bring this up because it's plausible and it's likely that as more people are being sent into space and spending longer periods of time there, it's more likely that they'll incur some kind of skin wound through bumping into something, anything like that. And so it's really of interest to sort of study the physiology of skin wound healing in this sort of space flight environment. Moving on to some background on wound healing in space, we decided to focus our efforts into 2 primary areas. The first one being microgravity, and the second one was hypoxia. So microgravity can directly impair tissue repair and wound healing through the various mechanical and biochemical factors, just as a result of, having a lack of gravity. And the second one is hypoxia. And hypoxia is essentially when the wound isn't getting enough oxygen. And that can also really impair wound healing. And the reason of interest is that these different spacesuit, spaceship microenvironments that these astronauts are undergoing, these could possibly induce some kind of hypoxia environment. And in fact, NASA stated in the future that the future exploration atmosphere during extravehicular activities, kind of like this, these will be at an atmosphere environment that would put the astronauts at a hypobaric hypoxia. And it's suggested that microgravity does impair cell migration. And we'll talk a bit more about what cell migration and what a migration assay really is. But the wound healing is characterized by a delayed lesser reepithelialization of the wound. And you can see on the right, I'd just like to focus your attention to the first 2 columns where we see a migration assay done on fibroblast cells. And basically, you can see the sort of wound boundary for control, which is like regular Earth's gravity, versus microgravity. And you can see that as each time point goes on, the wound area is a bit more for those in microgravity. And that suggests that these cells are migrating and closing up, healing more slowly as a result of being in microgravity. And the other half is that hypoxia inhibits wound healing due to insufficient oxygen delivery to damaged tissue. That's kind of just a known fact. So the goal of our studies and our experiments was to observe the effects of simulated microgravity in conjunction with hypoxia on fibroblast cell migration, which is an interesting endeavor because we're often seeing these studies that can prove that microgravity, like in isolation, can impair wound healing or just hypoxia. But we want to see, realistically, these astronauts are not experiencing one thing at a time, but they're probably, it's all at once. So that's what we were looking for. So I mentioned something earlier called a cell migration or migration assay. And basically, cell migration is referring to how these in vitro wounds are closing up over time. And we measure that using something called a scratch migration assay. And the way that we typically do it, so first I'll explain how we usually or traditionally do it, and then I'll explain kind of how we've adapted it to our needs. The way that we typically do it is we will plate cells onto something like a 12-well or 24-well plate. And once the cells are totally confluent, we can use something like a pipette tip to induce a wound or a kind of scratch. And that's what you're seeing outlined in blue here. This is what we call the wound boundary. And over time, as we image the cells, we can measure this wound closure by seeing in our images the wound boundary closing. And since we're doing space-related research, of course, we have to have a sort of way to simulate or model microgravity in our regular benchtop lab environment. And the way that this is typically done is with a fancy kind of equipment called a rotary cell culture system or a random positioning machine. And these are types of bioreactors that through some kind of spinning or rotation, they act to nullify or sort of eliminate the gravity vector that these cells would feel typically. And on the top right, you can see the rotary cell culture system. It's kind of a bioreactor that's fixed on the wall, and it'll spin the cells, and they're constantly in suspension moving. And the same, it works on a similar principle for the random positioning machine. It's a little bit more fancy in that it's rotating on 2 different axes, but the purpose is the same. The issue is that these types of equipment are very expensive, and there are a lot of logistical issues in terms of being able to perform the kind of experiments we want to do. If we want to do a migration assay, you have to be able to pin the cells down and kind of do a scratch on it. But if, like I said, in the rotary cell culture system, they're constantly spinning around and in suspension, that's not really possible, or we would have to kind of work around this in an awkward way. So we had to ask ourselves if there is a simpler, less expensive way, especially amidst the various restrictions we were under this year. And the answer is yes, and also that sometimes the simplest solution is the best. We devised something that we like to call the flask inversion model for simulating microgravity in vitro. And we call it the inversion model, but in the simplest sense, it's preconditioning the cells by plating them in a regular position. And then upon inducing the wound, we'll flip them upside down. So they've spent their whole time growing in this regular gravity situation, but once we scratch and we fill it all the way up with media so that there's no like kind of splashing or weird kind of shear happening, these cells are effectively experiencing this kind of microgravity in a crude, easy way. And the pros are that it's inexpensive, and this is easier to perform the scratch assay, at least compared to the other models I was mentioning earlier. And here's just a figure of how we perform the scratch assay on the flask. So you'll see I drew kind of a grid on each flask, and this is used to indicate the center of each flask. The way we perform migration assay with this kind of new model we're testing is we plated 200,000 cells per flask, and we allowed it to culture. Once it reached the appropriate confluency, we scratched it with a 1 ml pipette tip, and we filled it all the way up with media. After scratch and filling it up, we allowed it to culture, incubate in our experimental conditions. So our experimental conditions are either the inverted model or just regular, normal. And we also did hypoxia versus normoxia. Hypoxia was 1% oxygen, and normoxia is just your standard 20% oxygen. And this ran for a 24-hour study. We imaged at 0, 18, 24 hours. Right, and here's an image of the normal. So this is normal. And here, this is our inverted, or basically is flipped upside down. And it seems like very simple, but this was more or less exactly what we were looking for, or this met our needs. And here's a figure of the initial wound area, and this is at time 0, right after you just made the scratch. Okay, moving on to the results. At 18 hours, cells in hypoxia exhibited greater percent wound closure than those in normoxia. And at 24 hours, hypoxia groups generally performed the best. And interestingly, we didn't observe any clear difference or trend between this inverted normal model we had going on. So in this figure here, you see in the orange and blue, these are hypoxia performing better than the gray and yellow normoxia. And at 24 hours, we say generally that these hypoxia groups are performing better. And if you were listening to what I was saying at the beginning, these results would be very confusing, because I spent the first half talking about why hypoxia and this flask inversion microgravity should really be impairing these wound healing responses. So what's the deal? And we think, well, firstly, based on the results, it suggested that cell wounds in hypoxia close more quickly than those in normoxia. And there's also seemingly no kind of trend in the inverted versus normal. And while this is seemingly counterintuitive, there was actually a lot of literature we ended up finding that found similarly that hypoxia can actually enhance skin wound healing. And the way that it does that is through something called hypoxia inducible factor 1. So short-term exposure to hypoxia can actually accelerate early wound healing, particularly in these fibroblasts. And what this HIF1 is, its function or purpose is it stimulates fibroblast migration, and it also upregulates the expression of growth factors and cytokines that are responsible for kind of kicking off this wound healing response. However, the catch is that to continue this wound healing, you need to later reoxygenate the wound. So this is really a short-term phenomena, and so we concluded that we needed to observe the cells for a longer period of time, especially because, you know, the hypoxia normoxia was only 1/2 of what we were interested in. The other half is this microgravity model. And since the data suggested no clear trend, we have to note that the results are largely affected by the initial wound size. So fibroblasts, for context, when you perform a migration assay on these cells, they're typically a 24-hour experiment, because just by the nature of how they close, they tend to close within 24 hours. So wounds that were too thin were more likely to close prior to seeing the full effects of the modeled microgravity. We concluded that this data wasn't really capturing the effect of our model. And so we decided that we needed to extend the observation period, or the duration of the study, to see what this model actually does. And the easiest way to do that, if we want to extend the observation, naturally we just increased the initial wound size. So we swapped out the 1 ml pipette and we used the 2 ml aspirating pipette. And this transformed our study from a 24-hour one to a 24 or 72-hour. It's actually ongoing right now. I have to take pictures of it later today. So it's still ongoing. And here's a comparison of the two initial areas. If you look on the left, this is the first iteration, and on the right is with this adaptation or revised model. And if you look carefully at the scale, the new model produces a much larger initial area. And the results are really quite interesting. So at the first 24 hours, we see a similar trend in terms of hypoxia versus normoxia, where hypoxia, particularly the hypoxia normal, is performing the best. But beyond the 24 hours, normoxia kind of starts to exhibit greater percent closure relative to the other groups. And interestingly, we are seeing in the first 24 hours, there's no noticeable trend again in the inverted normal configuration. But as time goes on, we see a greater difference between the modeled microgravity. We see the normoxia group that was in microgravity is performing much worse compared to those in the regular gravity. And this is kind of what we expected, because if we look at the literature again, we saw that firstly for hypoxia, we thought that this short-term exposure, this was only for the short term, and then beyond that, we needed to reoxygenate it to continue the wound healing. So beyond that 24 hours, we see that normoxia does start to perform better. And it was as we expected that as time went on, this model for microgravity does show more effect. So this was very promising, and the results are ongoing, like I said. For conclusions, we can conclude that hypoxia enhances early cell migration, but it later inhibits wound healing responses. And that our kind of simple model for microgravity does inhibit cell migration also later on beyond that 24-hour time point. And for future directions and future experiments, we plan to conduct similar studies using this kind of revised model, but on additional cell types. Okay, here are my references. Thank you all for listening. Sorry for technical difficulties at the beginning. Thank you.